Hello, my dear friends. Today, again, we are meeting for one another lecture that is of paper number 503 Molecular Biology and Bioengineering. In today's lecture, we are going to discuss regarding the models of DNA replication. Now, in this topic, we will be discussing about the three basic models how DNA replicates and which is the correct model and why it is the correct model that we are going to discuss in this episode. So again, I welcome you all. So let's begin. Okay, so to start with, what is DNA? We all are aware DNA is deoxyribose nucleic acid. Now, why it is important? Because it is the genetic material of all, most of the majority of the organisms present on this planet Earth. Only exceptions are few plant viruses where RNA is termed or present as the genetic material. Now, when a cell divides, it is important that each daughter cell receives an identical copy of the DNA. Now, why it is important that it receives identical copy? Because if it does not represents or receives identical copies, it is very difficult for an organism to regenerate or reproduce in the next generation. This is accomplished by the process called DNA replication. Replication means what? Replication means a kind of a copy from one generation to another generation. The replication of DNA occurs during the synthesis phase or S phase. Now, if you have already studied cell cycle, you will be aware about this S phase. If not, please go through your previous year syllabus, study mitosis and meiosis. It is very helpful for you to understand the further topics. Now, next. DNA replication is the fundamental process occurring in all living organisms to copy their DNA. Whenever we are saying the words photocopy, that means exact copy. So DNA replication is one of the process where exact identical copies are made. Now, the process is called replication. In that sense, strand of double-stranded DNA serves as the template strand for reproduction of the complementary strand. Now, what is template strand and what is complementary strand? A strand which is used as the one, and the one which copy is made is complementary strand, and the one which is used as a reference is termed as template strand. Because of the complementarity of the two strands, having one strand means that is possible to recreate the another strand. Now, these sentence I'll be explaining you in the next few slides, right? So let's go further. Now, which are the general features of DNA replication? So various features are there. Here is a list of few of the features. Like DNA replication is semi-conservative. The first point itself says that why it is semi-conservative. That is what we are going to study in the further slides. Next is it is bi-directional process. It is never unidirectional. DNA replication is always bi-directional process. I guess in standard 11th and 12th, you have already studied some part of the DNA replication. Next is it proceeds from a specific point called origin of replication, or ORIC. Many a times we have come across this word, that origin of replication means the point from where the replication is starting. Next is it proceeds in 5' end to 3' end. Always it starts from 5' to 3' end. It occurs with the high degree of fidelity. Why high degree of fidelity? Because it is always occurring in a continuous manner. Next, it is multi-enzymatic process. Various, more than 10 enzymes are used in the process of DNA replication. Now, there is some difference between the process of DNA replication in prokaryotes and eukaryotes. Especially in eukaryotes, three major things are there: initiation, elongation, and termination. 3 steps replication is divided into. But here we are not going further in the process of replication because it will take one more lecture for you to understand the whole process of replication. Today we are just overviewing few of the models how DNA is replicating. So let's begin with the models of DNA replication. Now, first is a semi-conservative model. Now, what do you mean by word semi? Semi means half. Conservative means half of the things will be conserving. Conserve means to preserve. So, in this semi-conservative model, the two parental strands separate and each makes a copy of itself. What is explained over here? The two parental strands separates and makes a copy of itself. Now this can be studied in this particular diagram. Two parental strands makes copy. After one round of replication, the two daughter molecules each comprise one old and one new strand. Again, I'm repeating that after one round of replication, so once replication is done, the two daughter molecules which have been formed, they are comprising one of the previous, that is old strand, and another that is of new strand. See, this is first round of replication. So after this, what is happening? One is the older one, that is the strange dark blue in color, and one is the newer one, that is light blue in color, that is sky blue in color. So after one round of replication, note that after two rounds, two of the DNA molecules consist only of new material, while the other two contains one old and one new. So whenever you are doing mathematics, so one is divided into two. Now two, when they are further divided, now what will be the scenario? You can observe over here. See, one is having old strand, one is having old strand, but the rest of the two both are having new. So this you can practically even work out in your books, so you will be able to understand then why it is such, right? So one, what the semi-conservative model says, that some part of the DNA, suppose this is from the father and this is from the mother, then you will see that after some generation, some part of the DNA is always conserved. Because of this, it is termed as semi-conservative model. So we are not either 100% similar to father or mother, or either 50% similar to father or mother, but we are having some of the characteristics either of the father or of the mother, because of this semi-conservative nature. I hope you all are able to understand what I want to explain. See, whenever the replication takes place, from the one parental strand, two will be formed, two daughter strands will be formed, where one will be having completely new strand and another will be old strand. But now when further replication is done, from this, one will be completely new and one will be having some portion reserved or conserved. So this is semi-conservative model of DNA replication. Going further, next model is conservative model. In the conservative model, the parental molecule directs synthesis of an entirely new double-stranded molecule, such that one round of replication, one molecule is conserved as two old strands. This is repeated in the second round. Now I know you are not able to understand what I want to say, but if you see the figure, it will be clear. Now, suppose the DNA replication is in the form of conservative model. What will be the scenario over here? In this conservative model, what is explained, that when this is going to replicate, one will be totally conserved and another will be totally new. So if suppose you are considering this is something extraordinary, or you can say extra and ordinary. Why? Because this is same like this, identical like this, but the another molecule which is strand, which is substituted or which is synthesized, is completely new. Now when further you are going to replicate this, what will be the scenario? Again, one will be like this, another will be completely new strand. But when this completely new strand will be replicated, you will see that there is no similarity with the original parent. So in conservative model, only one is having the characteristics same like parents, other will be completely new. So if you see or think on this model, you will feel that it is not possible for the living organisms, right? Moving forward to the third model, that is dispersive. What itself is, it is dispersed in various directions. Material in the two parental strand is distributed more or less randomly between two daughter molecules. What is explained over here? It is distributed more or less randomly between two daughter molecules. In the model shown here, old material is distributed symmetrically between the two daughter molecules. Other distributions are also possible. Now see, what happens if the dispersive model is to be followed in the replication? Some part will be old, some part will be new. So it is totally random. What will be new and what will be old, we cannot predict. Now, when further this two parental strand will replicate, what will happen? More of the new parts and rest, very less residues will be there of the parental generation. Now, this is one of the case. Now, it can be in any zigzag manner. You cannot say that, no, this part is of first strand, this part is of second strand. So it is dispersive model. Thus, we can say that only the semi-conservative model fits into our mind. Why? Because it is intuitively appealing model. Because separation of two strands provides two templates. One will be older one will be newer one. Each of which carries all the information of the original molecule. Now, if this is carrying all the information of the original molecules, means whenever you are taking, so there is some sayings in Gujarati that 7 generations, there are some things which are going on and on and on till 7 generations. Why? So now you can correlate that thing over here. Because it is semi-conservative, some of the things will be preserved. Some things you inherit from your father, then some from your grandmother, then your more grandfathers, or something. So till 7 it will be like that generally. Why? It also turns out to be correct one. This was explained by the two scientists Messelson and Stahl in very few lectures. We are going to discuss about this Messelson and Stahl's experiment. We are going to understand that why DNA is considered to be the genetic material. So these all things will be studying in the upcoming lectures. But for now, I hope you all are aware about these three models. It is very, very easy to understand. You just have to remember three words: semi-conservative, conservative, and dispersive. Let us take a summary or a recap, so it will be easier for you to understand what these three models wants to say. See, in semi-conservative model, parental generation after first replication cycle, after second replication cycle. Here what you will see, one strand is conserved and one is newer one. Same things is occurring over here. If you consider conservative model, one is same like parent, one is completely new. So completely new, all new things will be present, but over here it is never such. Here, if you can take the case or scenario, all the things, one will be completely like the old parent, one will be completely new. And if you discuss about the dispersive model, here after the parental replication, you will see that after first cycle, few of the things are here. See, suppose you consider this pink parental, you will see some pink is over here, but it is not never possible that same things will be coming over here. So it is dispersive, it is scattered in manner. So after first replication cycle, you will get something which will be similar to this. You can correlate to semi-conservative and dispersive. But after second replication cycle, you will be able to understand that it is never possible in living organisms. Why? Because if it is dispersed, then we cannot say that which gene is responsible for which characteristics. So this is in short, summarized in short. Please study various reference books. You will be able to understand it very easily. You can use this video lecture and note it down in your books, and you can understand it by yourself. That will be much easier and better way. I hope you all are preparing the respective notes of every lecture which is being taken and conducted, because at the end of the semester it will be difficult for you to understand. It is not possible to study and view all the videos at a time. So start preparing your own notes, start making few points. You can highlight it, you can make points, you can do it by your own self, you can share it with your friends, right? So with this, today I want to conclude my session. These are few of the references where the materials is being collected. These are the list of your reference books which you need to constantly study. Links will be there in the net, in the Google. Search it, find it out, take an ebook and start reading it. Keep it in your cell phone, so every now and then you can just go through it, right? So with this, Let us conclude our lecture. I hope you all are safe at home. Don't roam unnecessarily and start studying, start working hard, because this is semester 5. Genetics, one of the very important subjects to be studied. Thank you so much all for your kind attention. Hope you are liking the videos, and if you are having any queries, you can contact the teacher. Thank you so much.